This video is about T lymphocytes, T cell development and survival, and this particular study note is developed by EPG Pathshala, MHRD team. So I would like to express my sincere gratitude to the development team. Thank you so much. Now let's learn. Immune system helps us to fight with pathogens that we encounter every time. T cells are central players of our immune system. Hence, a thorough study of T cell development is very important, that it will help us to know how T cells acquire its receptor, T cell receptor, with the help of which it recognizes antigen presented on MHC molecules, how T cells are educated about self and non-self moieties, how it attains self-tolerance, how it develops strategies and manage acceptance of organ transplants. The knowledge and understanding of development and TCR expression pattern of T cell, which may help to devise strategies to transfer T cells in immune compromised patients, HIV. So, introduction: T cells at a glance. The immune system is made up of two components: innate and adaptive immunity. Innate immunity is non-specific but very crucial for defense of the body. Adaptive immunity is what we acquire during our lifetime due to exposure of various pathogens. Adaptive immunity is very specific and not only fight and neutralizes the pathogens, but also remembers the pathogen so that reinfection with same pathogen leads to faster response. Though functionally different, both innate and adaptive immunity works hand in hand for the optimal immune responses. Adaptive immunity is driven by B and T cells, which performs distinguished functions during microbial invasions. T cells play an important role in mounting effective response against pathogens and cancers, whereas B cells are the main antibody secreting cells. The important structural and functional attributes of T cells are as follows. T cells are named so as they mature in thymus. Among several B and other cells, T cells constitute major proportion of blood cells. They play an important role in defense as they recognize the antigen presented by class 1 and 2 major histocompatible complex molecules on antigen presenting cells or APCs. The distinguished characteristic of T cell is that they can recognize antigens displayed on MHC molecule, same as of host's MHC from which they are isolated. T cells demonstrate self-MHC restriction. The distinguished characteristic of T cell is that they can recognize antigens displayed on MHC molecules, same as of host's MHC from which they are isolated. T cells demonstrate self-MHC restriction. Mostly T cells don't activate in response to the self-antigens of host from which they are isolated. Hence, T cells demonstrate self-tolerance. T cells have T cell receptor, which with the help of which they recognize antigen on self-MHC molecule. Along with TCR, they have CD3 molecule, which helps in transducing downstream signaling after antigen binding and leads to activation. Most of T cells have TCR with alpha and beta genes, alpha beta T cells. Few are present with gamma and delta genes, T chain cells. The gamma delta T cells differ from alpha beta T cells in antigen recognition as they have limited antigen diversity and recognize antigen presented by CD1 molecules, MHC like molecules. Alpha beta T cells also have CD4 or CD8 core receptors, which further helps in antigen recognition. In fact, CD4 displaying T cells, also known as T helper cells, can interact with antigen presented on MHC class 2 molecules. So, CD4 T cells are class 2 MHC restricted, while CD8 T cells, also known as T lymphocytes, T cytotoxic cells, are class 1 MHC restricted. CD4 T cells MHC2, CD8 MHC class MHC1 restricted. The alpha delta T cells don't have CD4 as well as CD8 co-receptors. Then T cell development, origin in bone marrow. T cells, like other blood cells, originate from multipotent hematopoietic stem cells in the bone marrow. Hematopoietic cells differentiate into myeloid and lymphoid progenitor cells under the influence of lineage-specific transcriptional factor. The lymphoid progenitor cells further differentiate into T cell progenitor cell among many other cells. You could see this blood stem cell, myeloid stem cell, lymphoid stem cell, lymphoblast, plasma cell, T lymphocyte, natural killer cell, B lymphocyte. Then myeloid stem cell, red blood cell, myeloblast, eosinophil, granulocyte, basophil, neutrophil, and along with that platelet is also there. So development in thymus, movement and maturation. T cell progenitor cells migrate to thymus at about day 11 of gestation in mice and in the 8th to 9th week of gestation in humans. Further maturation of T cell progenitor cells happen in thymus. The developing T cells are also known as thymocytes. During maturation, thymocytes interact with stromal cells of thymus such as epithelial cells, dendritic cells, and macrophages, and their interaction contribute to the growth and maturation of thymocytes. The major changes include arrangement of the germline TCR gene followed by acquirement of functional TCR gene, then expression of specific cell surface membrane markers such as CD4 and CD8. They get trained by thymic education. This includes recognition of MHC molecules by thymic stromal cells, thus become self-MHC restricted and non-reactive to self-antigens, thus becoming self-tolerance. This training is popularly known as thymic selection. The maturation steps can be divided into various steps like T cell progenitor cells proliferation and differentiation. Basically, T cell progenitor that arrives at thymus is completely different from mature T cell and can be stained for the following molecules. CKIT, receptor for stem cell growth factor, CD44, adhesion molecule involved in homing, no rearrangement of TCR gene and lacks TCR and CD3 complex, no expression of CD4 or CD8 co-repressors, CD4 and CD8 negative or double negative DN1. T cell progenitor cells generally enter at the outer cortex of thymus and slowly proliferate for about 3 weeks. During proliferation, it starts expressing CD25 alpha chain of IL-2 receptor. At this stage, the cell is DN2, double negative 2. Then it differentiates into DN3 under the influence of thymic stromal cells. During differentiation, it shows reduced expression of c-KIT and CD44. So CD25 alpha chain of IL-2 receptor expression. Switch on the expression of RAG1 and RAG2 gene expression, the product of which is required for rearrangement of TCR gene. Initiate rearrangement for beta chain genes of TCR. Some show rearrangement of gamma and delta chain genes. A DN3 cell which successfully completed rearranging beta chain survives and loses CD24 plus and progress to DN4 stage, while the cell that failed to rearrange beta chain locus remain at DN3, CD44 and CD25 plus stage and soon die by apoptosis. So here you could see some shows rearrangement of gamma and delta chain genes. A DN3 cell with successfully completed rearranged beta chain survives and loses CD24 plus and progresses to DN4 stage, while the cells that fail to rearrange beta chain locus remain at DN3, CD44 and CD25 plus stage and soon die by apoptosis. Precursor NK DN1, CD25, CD44, alpha delta TCR, DN1 to DN2, DN3, DN4, pre-T cells, DN pre-TCR, CD4 plus, CD8 plus, DP. Different stages of thymocytes. T cell progenitor goes through various double negative stages, DN1 to DN4. During proliferation, it passes through DN1 and DN2 stage. DN2 specializes to form DN3 with reduced expression of CD44, increased expression of CD25 and RAG genes. DN3 cell which successfully completed beta chain downregulates CN25 expression DN4 stage, which further differentiates into pre-T cell where beta chain combine with pre-alpha chain to form double negative pre-T cells with pre-alpha beta TCR receptor. Then formation of pre-T cells. Formation of pre-T cells. The beta chain combines with 33 kilodalton glycoprotein known as pre-T alpha chain and associates with the CD3 group to form complex called as pre-T cell receptor or pre-TCR. Studies indicate that the pre-TCR complex got involved in signal transduction in ligand-independent manner by CD3, which in turn helps in the following process. Proliferation of pre-T cell suppresses further rearrangement of beta chain genes, induce expression of CD4 and CD8 membrane molecules, permit for rearrangement of alpha chain genes. Pre-TCR, pre-T-alpha, TCR beta. Cell becomes permissive for TCR alpha chain locus arrangement, stimulates expression of CD4 and CD8 co-repressor, stimulates proliferation, stops additional TCR beta chain locus arrangements, allelic exclusion. So pre-T cells with pre-TCR, it interact with thymic stromal cells. TCR complex initiate downstream signaling via CD3, which in turn help in formation of double positive pro-T cells with rearranged alpha and beta TCR genes. The pre-T cell proliferate in rapid manner, although they show increased expression of RAG1 and RAG2 genes, but in proliferating cells, RAG2 proteins degrade rapidly. Only cells with rearranged beta chain proliferate and make clones of itself. When the proliferative phase ends, the alpha chain genes show rearrangement. In different clones, different alpha chain gene undergoes rearrangement, which further adds to diversity of rearranged TCR genes. The pro-T cells with rearranged alpha and beta chain are also both CD4 and CD8 positive, hence double positive DP cells. Then thymic education and formation of single positive T cells. An enormous diversity of approximately 10 raised to 15 for A alpha beta chain is generated due to random gene arrangement with TCR germline DNA of alpha and beta chain gene. The double positive pro T cells with successfully rearranged TCR receptor interact with stromal cells via rearranged TCR complex. Their interaction becomes a deciding factor for the fate of T cells and results in formation of single positive T cells which are of self-MHC restricted and self-tolerant. Thymic selection. Positive selection ensures self-MHC restriction. During this process, only those double positive thymocytes with rearranged TCR genes are selected that interact with the MHC molecule displayed on cells of stromal cells of thymus. The thymocytes which don't react are eliminated by apoptosis, which leads to generation of thymocytes which are self-MHC restricted. Positive selection happens in the cortical region of thymus. The TCR of thymocytes interact with MHC molecules, class 1 and class 2 molecules displayed on surface of cortical epithelial cells. Positive selection, first negative selection. Then depending upon, this is positive selection. Okay, positive selection ensures self-MHC restriction. Positive selection happens in the cortical region of thymus. The TCR of thymocytes interact with MHC molecule, class 1 and class 2 molecules displayed on surface of cortical epithelial cells. Depending upon interaction between thymocytes and epithelial cells, it generates either single positive MHC2 restricted CD4 plus CD8 plus or MHC1 restricted CD4 minus CD8 plus cells. Then negative selection. So here we will be choosing, I'm talking about positive selection. The thymocytes which don't interact are eliminated. Or those which are reacting with thymocytes are selected. Negative selection ensures self-tolerance. During negative selection, those thymocytes are eliminated which interact very strongly with the MHC molecule alone or with self-antigens displayed on self-MHC molecules. It leads to the generation of thymocytes which are not specific against self-antigens. Positive selection happens in the medullary region of thymus where thymic dendritic cells and macrophages interact with positively selected thymocytes or self-MHC restricted thymocytes, and those thymocytes which interact too strongly with MHC molecules alone and with self-antigen presented on self-MHC molecules get a negative death signal, while others with low affinity receptor for self-antigen and self-MHC continue to mature. Okay, thymocyte from bone marrow, rearrangement of TCR genes, CD8, CD3, T cell receptor, CD4, immune thymocyte, positive selection of cells whose receptor binds MHC molecule, class 1 and class 2 MHC molecule, death by apoptosis of cells that do not interact with MHC molecule. Then thymic cortex, negative selection and death of cells with high affinity receptors for self-MHC or self-MHC plus self-antigen, CD4 plus CD8 plus macrophage and dendritic cell. Thymic selection of double positive pro-T cells. Thymic selection of double positive pro-T cells. Positive selection occurs for the thymocytes which interact with peptide MHC complex on thymic epithelial cells and get survived, while cells which do not interact die by apoptosis. Negative selection occurs for positively selected thymocytes, the thymocytes which interact very strongly with MHC complex alone or with self-peptide MHC. Wait a minute. Positive selection occurs for the thymocytes which interact with peptide MHC complex on thymic epithelial cells and get survived, while cells which does not interact die by apoptosis. Negative selection occurs for positively selected thymocytes, the thymocytes which interact very strongly with MHC complex alone or with self-peptide MHC complex displayed on the surface of dendritic cells and macrophages of thymus gets die by apoptosis, while others get mature into single positive CD4 plus and single positive CD8 plus cells. Then experimental evidence for essential elements for selection. The early evidence for the central role of thymus is provided by chimeric experiments done by Ziegler-Nagel and his colleagues. They took a thymectomized and irradiated strain of mice F1 with MHC haplotype, and mice were implanted with thymus graft from strain B with MHC haplotype H2B. The thymus was irradiated before transplantation to remove any mature T cell from thymus. The immune system as recipient mice was reconstituted by giving intravenous injection of bone marrow stem cells from F1 mice strain. It was noted that the T cell progenitor from bone marrow would migrate to B type thymus that expressed B type MHC on its stromal cells. Now the question was, would T cells be restricted to MHC of thymus graft or MHC of recipient mice strain? To answer this, mice was infected with LCM virus. The spleen containing T cells was taken from infected mice. Spleen cells were incubated with target cells infected with LCM virus from strain A and strain B. They observed that T cells could kill only target cells from strain B. They don't respond to target cells from strain A. It implies that they recognize MHC of strain B, which is similar to implanted thymus or T cells were MHC B type specific. It indicates that thymic MHC haplotype plays essential role in restricting MHC range to which T cells can respond. While in control studies after infection with LCM virus, T cells can kill infected target cells of both the strains. Further, direct evidence for interaction of class 1 and class 2 MHC molecules displayed on thymic stromal cells with TCR of thymocytes during positive selection is provided by experimental studies in knockout mice. Class 1 MHC knockout mice showed a normal distribution of double negative, double positive cells and single positive CD4 plus cells, but lacked a population of single positive CD8 plus cells. Class 2 MHC deficient mice had double negative, double positive and single positive CD8 plus cells, but lacked a population of single positive CD4 plus cells. Evidence for elimination of thymocytes reactive for self-antigen displayed on self-MHC molecule negative selection came from the following transgenic experimental studies. In this experiment, alpha-beta rearranged TCR genes were taken from CD8 plus T cells, clone specific for antigen HY peptide displayed on class 1 MHC molecule of H2B type. The HY peptide is an antigen that is encoded by Y chromosome in male, thereby functioning as self-antigen for male mice. The alpha-beta rearranged TCR gene were H2B MHC specific displaying HY peptide. The genes were inserted into fertilized egg of strain with MHC haplotype 2AB H2B, same MHC haplotype to which TCR genes are restricted. When the fate of T cells were observed in male and female transgenic mice, it was found that female transgenic where HY peptide is not a self-antigen had double negative, double positive, single positive CD4 plus and single positive CD8 plus. While male transgenic where HY peptide is a single antigen, it showed the population of double negative, double positive, single positive CD4 plus, but lacked single positive CD8 plus, thereby indicating that HY specific CD8 plus cells did not survive and underwent deletion. Thus, the experiment provides a striking example for negative selection of T cells recognizing self-antigen. Then also the role of peptides on class 1 and class 2 molecules during interaction with thymocytes are provided by TAP1 knockout mice studies. TAP1 is involved in transporting peptides from cytoplasm to endoplasmic reticulum from where they got attached to cleft of class 1 MHC molecules. In TAP1 knockout mice, peptides couldn't bind to class 1 MHC molecules and class 1 MHC molecules were not displayed on the surface. These mice lacked the population of single positive CD8 plus cells. However, when exogenous peptide were given to organ culture, class 1 MHC molecules appeared on the surface and CD8 plus cells population were restored. So, mechanism of thymic selection and a single positive cells formation. During positive selection, the interaction between TCR of thymocytes and peptide MHC molecules on stromal cells lead to survival of cells with self-MHC restriction. During negative selection, the interaction leads to thymocytes elimination. So, both the selection would lead to very poor outcome for T cells. However, interestingly, it is not the case. There is still a way that significant number of T cells are generated. There are two hypotheses proposed to distinguish interaction between positive and negative selection. Differential avidity hypothesis. The interaction between TCR MHC peptide complex in thymus determines distinct signal for selection. For example, no interaction leads to no signal, which result in death of thymocytes. Weak interaction leads to less signal required for survival of thymocytes, positive selection. Very strong interaction leads to more signal, which results in elimination or negative selection. Then differential signaling hypothesis, the interaction between TCR and MHC peptide complex involve different sets of peptides. Agonist peptide delivers very strong signal which lead to negative selection. Agonist peptide delivers partial signal which favors positive selection. The next question arises regarding the identity of interaction which lead to generation of single positive cells from double positive cells. There are several hypotheses proposed. Instructive model hypothesis states that interaction between peptide class 1 MHC molecule and double positive thymocytes instructs the cells to differentiate into single positive CD8 plus cells, whereas interaction between peptide class 2 MHC molecules and thymocytes instruct the cell to differentiate into single positive CD4 plus cells. Then stochastic model hypothesis believes that there is random downregulation of CD8 or CD4 co-receptor in double positive cells. The cells with high CD8 and low CD4 are able to interact with class 1 MHC molecule and get survive if not undergo apoptosis. Similarly, cells with high CD4 and low CD8 are able to interact with class 2 MHC molecules and get survive if not undergo apoptosis. The mature cell positive class 2 MHC restricted CD4 plus, also known as helper cells TH cells, and single positive class 1 MHC restricted CD8 plus, also known as cytotoxic cells TC cells, which have the ability for self-tolerance are generated. The mature T cells move to the blood and reach secondary lymphoid organs where they interact with antigens. Now let's have a summary. To summarize, during maturation and proliferation stage, large number of T cell repertoire are generated with random combination of alpha and beta genes or gamma and delta genes with enormous specificity for diverse number of antigens, self or non-self. The adverse alpha beta T cells repertoire undergoes stringent rounds of selection where MHC peptides complex on thymic stromal cells interact with TCR on thymocytes. The positive selection states only those T cells which can recognize particular MHC molecules displayed on stromal cells, restricting their range from MHC recognition. The negative selection makes sure that only those MHC restricted T cells survive which cannot recognize self antigens. These interactions lead to generation of mature single positive class 2 MHC restricted CD4 plus, also known as helper cell or TH cells. The single positive class 1 MHC restricted CD8 plus, also known as cytotoxic cells TC cells, the mature T cells then migrate to the blood and reach secondary lymphoid organs where they interact with antigens.